So I know what you came to Baltimore not thinking that you were going to do this or any of this, but I thought my hope is that you'll leave today's lecture thinking, well, that's kind of cool. Okay, that's my only expectation or hope. And I should say I put this title very quickly, and it says how cells migrate. I really should say it's how some cells migrate. And in particular, the type of cell that we're going to look at is something like this. What you're seeing here is a movie from before I was born, 1950s, originally done in 8mm film. This is blood. What you're seeing, the round things here are red blood cells. They don't move, they stick around there. The big thing that's moving around is a white blood cell, a particular type of white blood cell known as a neutrophil. And the little thing, there we go. So that little thing over here is the Staphylococcus aureus bacteria. And as you can see, the job of these neutrophils is to hunt that bacteria and kill it. Okay. And as the bacteria is trying to get away, the neutrophil follows it, moves those red blood cells out of the way and eventually crunches it and engulfs it. Okay. So there's a lot of different behaviors that we're seeing. One of them is that the amoeba is following the bacteria. So how does this is a single cell. How does it know where the bacteria is? It has to actually move. And the way you see the movement is by these shape changes, cell shape changes. It doesn't have a flagella or anything like that. And in the end, it has to engulf it and kill it. Okay. All those would be really interesting topics. What I'm going to primarily talk about today is that middle one. How does the cell actually move? We don't work directly with the white blood cells, the neutrophils. The collaborators I have in the School of Medicine look at this particular amoeba. It's called Dictyostelium discoideum, or Dicty to its friends. It is, it kind of looks a lot like the neutrophils. And in fact, it's safe to say that almost everything that we know about neutrophils moving first was discovered in this amoeba. Okay? Because if you actually look at the biochemistry of the single cell amoeba, it's almost identical to that of us humans. So if that doesn't make you humble, I don't know what. Just to give you a sense of scale, how big are these cells? Well, there's about 10 microns across. Okay, and this movie is very much sped up. A cell like this moves about a body length a minute. Okay, so what you're seeing is several minutes worth. The thing that you see move around is a micropipette, and it has a particular chemical. And what the cell does, it has these receptors that are specific to this chemical, and they have the receptors all across the cell. But somehow the single cell amoeba is smart enough to actually compare occupancy of receptors across from one end to the other and use this to move. Okay, and it's extremely sensitive. You can have as little as of a 2% difference in concentration of the outside chemical between front and back, and the cell will actually go in the right direction. Okay? It doesn't do this perfectly in the sense that it is a random walk, but it's a biased random walk to get there. So just let me tell you a little bit, since I'm going to focus on how cells move, let me explain to you how it moves. And these cells use what's known as actin polymerization. What is actin? Actin is a protein. It is found in several states. I like to compare it to Legos because it's something I understand more than biology. You have these globular proteins, okay, and they're throughout the cell. Okay, think of your Lego blocks like that, and you can actually form and basically polymers of them, which is basically stacking all these globules together. Okay. And what those do, and here's an electron microscope picture of a very dense set of these filaments, is that they push against the membrane. When you stack up a new block at the end, you make it longer, and so that pushes the membrane. Okay? And as you do that, the shape of the cell changes. Okay? Now it's not quite Lego, not surprisingly, in the sense that there's a preference that well, in fact, like Lego, there's a front and a back, or a top and a bottom. But where the addition of new monomers goes is preferentially done at the top. And the other thing that happens is that they preferentially fall from the bottom. Okay, so you get this treadmilling effect where the thing doesn't necessarily grow in length, but it's growing in length towards out of the membrane. And because you can anchor it in this jungle of filaments, you actually get some force produced. Okay, and that's what you see over here. Again, this is just a single dictyostelium cell where we've done some image processing to show where the extensions happen and also where retractions happen. Okay, and if you, I'll run this movie again, but you see that once in a while, what you get is that where this pushing is come will actually split in two. Okay, these extensions are known as pseudopods, which is Greek for false feet, because they look like feet, but they're not feet. And so what you're seeing is known as a pseudopod split. And this was known for a long time experimentally. And I'm going to show you a little bit why that happens. Okay, sorry. Okay, so questions that, when we started this, questions that we had is what leads to the creation of pseudopodia, or these pseudopods, and how does the external signal, okay, that chemical that we are applying with the micropipette, bias the movement of it. Okay, now I'm going to show you, when I write a hint to the answer, I'm going to show you how we get information from cells. In this case, it's mostly microscopy. Now, there are techniques by which you can make proteins be fluorescent. And normally, if for example, if we were to tag actin, make it fluorescent, what would happen is that there's so much actin that the whole cell would be fluorescent. Okay, so that's not terribly useful. And basically, that's the way typical microscopes work. Okay, you shine a laser light, excites the fluorophore, it shines bright, you recover with CCD camera, and you see where the proteins are. Okay, but if you did this again, the whole thing would light up. So what they do is the microscopists use what's basically internal reflection. Instead of shining the laser at a 90-degree angle to the bottom of the cell, what they do is they shine it at basically the angle of incidence. Okay? So what happens is you get reflection from it. None of the light goes through it, but that's not 100% true. What you get is this very small, what they're known as evanescent wave, okay, where you actually excite the fluorophores that are very close to the bottom of the cover slip. Okay, and how close? In the order of 50 to 100 nanometers. Okay, so any of the fluorescent proteins you see are right on the membrane. Okay, and so that's what you're going to see. And if you do this with actin polymerization, you get something that looks like this. So remember, I'm looking at the bottom of the cell. So you see essentially propagating waves, okay? And they're happening at the very bottom, moving sideways. And the waves are happening wherever new actin filaments are being formed, okay? So you see these, it looks like you've thrown a pebble in the pond, except that when two waves hit each other, what happens? They actually annihilate, okay? So they're destructive waves. Couple things to note about this. One of them is that in a moving cell, those waves happen towards the front. Okay, so they're basically pushing the cell. Okay. The other thing is that it has nothing to do with that external source. This is the cell where they've taken away the receptor. It has none of those receptors for chemicals, and you still see the same sort of wave pattern. Okay, and just to show you how, I mentioned amoeba, the dicty amoeba and neutrophils. The first two are neutrophils, the third one's amoeba. So really, you can't really tell the difference from these movies. Okay, now, if you're a biophysicist and you see these waves annihilate each other, you know what to look for, and that would be what's referred to an excitable medium. Excitable systems goes back to the work of Huxley and Hopkins. They were looking at how axon nerve propagation happens. And they had this beautiful biophysical model of how action potentials move. Okay, and I won't go through it, but a more useful model to sort of a more parsimonious model of how this works is due to two sets of people. Richard Fitzhugh, who actually got his PhD here in biophysics in the 1950s, and a trio of Japanese experimentalists who were actually building an electronic circuit to make these action potentials, okay? And for some reason, we only ever remember Nagumo, who was the first author of the paper, but there were two other ones. And the notion that they came up with is very simple, what's referred to now as an activator-inhibitor system, okay? Where you have two processes. I'm gonna variously change labels during the talk, but for now I'm going to say X and Y. X is an X is an activator. So it has this very strong positive feedback. So if for some reason you trigger this, it will autocatalyze or increase its own production. It also increases the production of a Y, okay, which is known as the inhibitor. And the reason it's known as inhibitor is that Y provides negative feedback to the activation of X. Okay, and in the Fitzhugh-Nagumo model, the equations are very simple. Okay, it's a nonlinear equation for the activator where you have essentially a third order polynomial with a root at 0, a root at 1, and a root somewhere between the two of them. The minus y here tells you that there's the negative feedback. And then the equations for the inhibitor are completely linear. Now, if you are familiar with dynamical systems, the way to analyze a system like this is to to find the null lines, which are the lines in state space where the changes are 0. So x dot equals 0 and y dot equals 0. Okay, and sure enough, what you get is for the x dot equals 0 equation, you get a cubic polynomial, and for the inhibitor, you get a straight line. And so in the dotted lines here, I've plotted this out. Okay, and I've chosen the parameters in such a way that there's only one intersection, which happens to the left of the minimum of the cubic polynomial. Okay. Now, one thing I didn't mention about the inhibitor, but it was in the previous slide, is that there's an epsilon which regulates the speed of the dynamics of that inhibitor. And so the way that epsilon works is that it's usually a small number, so that any changes in the inhibitor tend to be much slower. Now, what that means in terms of dynamics is that any movement, if you're in a point away from the null lines or away from the equilibrium, is that any changes are going to be mostly horizontal. Okay? Now, the single equilibrium point is stable. Okay? And so a small perturbation about this is just going to return to it. Maybe not quickly, but it's going to return. It's not going to deviate from there fast enough. But if you somehow manage to perturb the system so that you reach, you start at point B, this change in the time scale separation means that you're going to rush out to the right. Okay, and that's the effect of the positive feedback, right? You've created slightly more X than there is at equilibrium. X builds up because of positive feedback, and it rushes out to this point C. At that point, you have crossed the null line, and so now you want to return, but you can't return right away because of the cubic null line. And so what you do is you build up slowly inhibitor, so that eventually you reach this point. At this point, you really can rush back, so you rush back to the left, and eventually slowly return to A. And what in the time domain, what these curves look like are this. Okay, so you can have fluctuations near the equilibrium. If for some reason we reach point B, you rush out, you increase very dramatically. In doing those, you're building inhibitor. Once you have sufficient inhibitor, you actually start dropping by. Eventually, you jump over here, and you return to that. Okay. If you add diffusion, okay, to these molecules, assuming they're molecules, then what you end up having are waves. Okay, and I'm not going, this is explanation waves for biologists who any math is a hurdle. But the idea is that the green circles are activated proteins they're diffusing around, and from the positive feedback is going to, they're going to move over here, and they're going to activate more of those molecules. Okay, and so you see propagation in that sense, but they're also turning on some of the red inhibitors, and they are diffusing as well, and so they're going to prevent the wave moving, in this case, to the left. Okay, and so you get propagation that goes to the right. Okay, just to show you in terms of simulation, this is a simulation of a Fitzgerald system where I've added noise to the activator. So you see a lot of changes, but every once in a while you cross that threshold for activation, and what you get are waves. And I don't know if you noticed, but when they hit each other, they sure enough kill each other. Okay. It turns out that you can build that positive feedback using a double negative feedback. Okay, so in this case, I changed my notation a little bit, but X, I've called it an F. An F here stands for front, because it turns out that in cells, we think of this system as related to the front. It inhibits a back state, and the back state inhibits F. So that little feedback there creates that positive feedback. And when you simulate this, I have this as the front, the back, and the refractory. You see that, in fact, it looks exactly the same. The interesting thing about this is that the refractory, refractory is what I called inhibitor before, sometimes looks like the front, sometimes looks like the back. Okay, so experimentally, if that's all you're looking at, it's really hard to tell with that protein whether it's one or the other. However, it's in terms of front and back, what you see is this division. Okay, so the space looks like there's, it's either front or back. And here's another simulation. The other thing you can see is that if you're only looking at one, sometimes it looks like you have these what we call shadow waves, which are essentially a wave of blackness moving. Okay. Okay. So why do waves stop? This was interesting because it led us to actually try to perturb the system. So basically, the null lines that I showed you look like this, and the equilibrium point is where the two intersect. And you can see that if you look at the cubic null line, you have a maximum and a minimum, and the intersection is somewhere between the two. Now, you can show mathematically that, in fact, the velocity of the wave will depend on the net area that is between these two areas. You take this as negative, you take this as positive, okay? And however much area you have, the wave will be proportional to that. So in this particular case, I have that intersection being at three very different places. At the top left, what you see is that the intersection basically has very little negative area. So that wave's going to move very quickly. If I change the level of inhibition so that it moves, the dotted line moves either up, you're going to have, a slower wave, and eventually you could have like a negative wave. Now, in practice, what happens is the inhibitor diffuses more quickly. And so what's happening over time in space is that level of inhibitor is actually moving up. And so you're going from this situation when a wave forms to this situation and eventually there. Okay? And of course, you do reach a point where the two areas cancel each other. And at that time, the wave stops. And this is in simulation of those three situations. Okay? What you're seeing here is what we call a kymograph. It is a picture of activity as a function of space and time. And so a wave was triggered at this point. Okay? And then it propagates in opposite directions. Remember, it doesn't come back the way it is. And a way of thinking of this is a forest fire, right? A forest fire consumes the wood, okay? And so there's no way that the fire can move backward because there's no wood there to burn, okay? So it's moving on either direction in a one-dimensional system, but you see that the curve, this is actually curving down, okay? And that's because the wave is getting slower, and eventually it reaches the point where the two areas are the same, at which point it stops propagating. Now, I'm going to tell you a little bit about triggering. Okay, so we tend to think of these the trigger of the wave as a stochastic process. So if you add a constant to the null line, it moves it up and down, and that changes the point of intersection. Now, if you think of that constant and you add some noise to it, you get this situation. Okay, so those are the same equations as before with some stochastic noise. Now, assume for a second that we are at a quasi-steady state. Remember, the equation for y is very slow relative to the equation for x. And so I'm going to set that equal to 0. So if epsilon equals 0, and solve for y, which is a straight line. I replace that into this equation, okay, and then write all this out as, you know, a cubic g of x plus a constant plus noise, okay, with variance sigma. Now you can basically I've reduced my system to a one-dimensional system, so I can now take the integral of the right-hand side and write this as a potential function. Okay, and what that potential function looks like is this. Okay, it's a double well potential, and we are operating at this equilibrium point. Okay, and you see the stability. Now, famous theory from Kramer argues as to how often if you add noise to this, how often you're going to cross the threshold and move to the other well. Okay, and basically equations given by that. It depends on, you know, the second derivative of the bottom of the well and at the top of the well, but primarily it depends on two things, or more accurately, the ratio of two things. One of them is how big that height is. Okay, I'm going to call that a threshold, and that relative to how much noise you have. Okay, so if you increase the variance of the noise, then, you know, more likely that you're going to cross it. If you make the well potential, the threshold really high, you're going to make it less often. I mean, and that's sort of makes sense. It's funny how great science almost always makes sense once they figure it out. Okay, and so what I've done here is just showing you what happens if I change the U naught, the constant part of the external signal, by a little bit. Okay, so blue would be sort of normal, and you go from red or green. And what you see is, of course, that not just changes things, you know, it changes things a little bit in terms of the null lines, but it really changes the relative height of those things. Okay, and so what's going to happen is that if you have the red and the green situation, you're going to cross that threshold a lot. So that means you're going to trigger a lot of systems. Okay, whereas if you raise it, obviously you do the opposite. Now, another way, we tend to think of the threshold as this distance between the top and the bottom. I should be careful, because if there are any experts here on excitable systems, tell you that's not a real threshold, and it's not. So sometimes call it a pseudo threshold. But because this is really only a threshold if epsilon equals 0. Okay, and epsilon's not equal to 0, or we wouldn't have an excitable system. But anyway, we take our standard model, which is a slightly different version of Fitz-Nagumo, but, you know, basically mathematically it's very similar, and look at different sensitivities for the different parameters of what that threshold is. And you see that, you know, A1 basically makes no difference to the threshold. Some of them, as you increase the parameter, the threshold gets bigger, like these three. Some of them get smaller. Okay. Now, why is this important? Well, long time ago, when we first discovered this, we made the following hypothesis: that in a chemotaxin cell, in a cell that's following a chemical gradient, what that chemical gradient does is it modulates the threshold of activation of actin polymerization. Okay, and it does so in such a way that it lowers the threshold at the front and raises it at the back. Okay, so that, you know, this is my analogy. Okay. So how many of you can jump that? Probably not many. I certainly couldn't. But if you lowered that, okay, more of us can be able to jump it. Okay, and therefore we're going to have more success. Where in case of the cell, I define success as creating a pseudoplate. Okay, and this is just showing you what happens if I change those same parameters that I showed you before. How often I get a spike or a threshold crossing. Okay, so by a small change, I can go from, you know, about 4 and however long this is to, well, basically almost a periodic wave. Okay, and now just to show you that this works, at least in theory, okay, we came up with a model by which we take a system where it looks at that chemical gradient, and I can't describe that, I don't have the time. But basically that triggers the reaction diffusion equations of this excitable system. And then we put that through a mechanical model of the cell. Okay, it's a viscoelastic description of how when you apply a force, the cell deforms, and simulated this using level set methods. Okay, and we try to be careful that this works. Okay, and this is what happens when we apply a gradient to the cell. Well, to the pseudo cell. Okay, you see that in fact it moves to the right. Okay, I'll run that again. Now, at this point, it's not moving anywhere, and the reason is I haven't applied the gradient yet. You'll see the whole cell turn red when I do apply the gradient. There it goes, and now you see. Now you obviously see a very stochastic behavior because it still depends on these threshold crossings, which are all stochastic. Now, another thing that you may see, let me see if I can stop it at the right moment, is that when a pseudopod forms, actually it does split into two. Well, I'm not so good at stopping it, but that does happen. And the reason is that when you create that wave tends to go in both directions. Okay. Now, why doesn't it turn into Y? Well, again, it's the stochastic nature of it. One of them tends to win out. And when this one stops, you cross in that direction. So the movement tends to look like a speed skater. Okay. Now, a couple things to note, and I don't have a ton of time, so I don't want to spend too long on this, but if you don't stimulate the cell, it pushes, right? Because you still get crossings of the threshold, okay? But, this is a simulation where we simulated 15 minutes, and what the cell did was they moved a lot like that, but it actually never got anywhere, okay? And that's a problem because we know that unstimulated cells still move around. Now, they move with no directional bias, but they do cover a big area because they're looking for food, right? Or bacteria if they're white blood cells. So there was something wrong with this. And what's wrong with this is that there is no memory to the system here, okay? Any pseudopod is just as likely to form anywhere, okay? Now the pseudopods in practice tend to be fairly small parts of an extension of the cell. And therefore, what happens is that if you push off in one direction, well, the next one complete the other direction. And so you see that the centroid moves around. The little dots in the middle, it's how the centroids move over time. But it never wanders very far away. Now, real cells do remember where they're going. Which, again, think about this. It's a one cell. There's no memory, or how does it generate memory? Sorry, this movie shows you that in fact what we described does happen occasionally, but not very often. So what we came up with is a concept that we call polarity, but other people use the word memory, which is the notion that you have two extra loops, two extra feedback loops. One of them is what we call a local positive feedback loop. Okay, and so what happens is that somehow the triggering of a pseudopod lowers the threshold of activation. Okay, and we tried that, and then the cells became super hyperactive. Okay, it's like they were blowing up. And so we realized we needed a second global negative feedback loop. And what that does is it actually basically balances the whole level of activity so that if you've lowered the threshold here, you've raised it everywhere else. Now, this is all internal, and the cell has to forget. Okay, so it's not it's a medium-lived memory. Okay, so that pseudopods extend over time in the same direction, but there's and that leads to actually a persistent motion, but it doesn't last forever. It lasts 6-7 minutes. And we got that number from real cells. So when you do that, and you add that module here, this is a movie of 5 of these pseudocells with no external gradient. Okay. But you see that they're actually moving away from the center. Okay, they're out there hunting for food. Now, all 5 of them went sort of in different directions, okay, but there is a persistent motion to them. okay? And that actually matches a lot of what we see. Now, how do we know? Oh, and I should point out that it does eventually forget. So this is, it may be one of these cells, maybe a different one, but what it did is it started moving to the right and it got bored. So it stopped. And I guess in that time that it stopped, there weren't enough threshold crossing. It started moving in the other direction. Okay. So on average, over time, over a lot of cells, you get an unbiased random movement, but over short periods of time, being 5-7 minutes, it is persistent. Now, how do we know that this is true in real cells? Well, I already told you that they move persistently. But one thing that you can do is you can bias the movement and then change the direction. What we were doing in that second movie that I showed you with a micropipette, right? Now, if there were no memory, what would happen is that the cell would move immediately into the new direction, okay? But if there's a memory, now it has conflicted signals, right? There's the immediate new gradient that it wants to move to, but there's also the memory of, I want to, where I should be moving. And so this is actually, you don't see a cell, but this is the outline of a real cell that was moving to the bottom. You don't see the old location of the needle, but then the needle was changed to where the star was. And what you see is that the movement is not a sudden in the new direction. What it does is it has this U-type turn. And this is a sort of recreation of that. There's a needle at the bottom. I just changed it to where the one you see, and you see that over time, as it forgets the old direction, it starts moving in the new one. Okay, but it does so through this gradual. This is I have another movie of that. Basically the same idea, but we're first of all, you actually see the trace, but I'll actually change the direction in various ways. So here, the green is going to eventually go in the right direction, but it takes a while, and ditto here. Okay, so that matches experimentally what we've seen. And you can't read this, but this point that this simple model actually was able to recreate all kinds of observed behaviors. Now, it's still a hypothesis, so how do we test it? Okay. Well, one other thing is that you look at properties of excitable networks. One of them is an all or nothing response. Okay, the notion that if you give it a small stimulus and a large stimulus, as long as you cross the threshold, okay, the response is basically the same. We showed that. Another one is that what's called a refractory period. That once you cross, you can't really trigger another one until you return. Okay, and I will show you this because it'll come up later. And the idea is here is that we stimulated the cell, stimulated a whole bunch of cells here, and then we waited some time to give it a second stimulus. So like this, where we're changing the location here. And you see that when it's really close, in fact, there is no second response. It's when as you wait longer and longer, the cell has recovered, and then you get a second response. And it took what, a minute before the cell gets back its original sensitivity. Inhibition, I mentioned this global inhibition. So one way of doing that is to give it a very high stimulus and then take things away from it. And sure enough, we did that. I remember this. Yes, so basically this was a high level of activity. So the model predicts that if you take away the stimulus, you're actually going to get a decrease in activity. And in fact, the experiment showed you basically the same thing. And the memory, I don't have a picture of that, so I'll skip that. Now, this is where I think things get a little more interesting if you're a control engineer. You can say, well, how can I break the system? Or more accurately, how can perturbations lead to different behavior? So what I've shown you is basically something that looks like that. Okay, you have the two lines intersecting at a stable equilibrium. Now you can see that if I change the slope of this, I could easily intersect at the high point. Okay, so then you would have a lot of activity, maybe. Okay, and you can ask, well, what's that going to look like? But at first, I'm going to show you another situation where I can thread the needle so that it intersects at several points. And in such a way, you can show that this is going through a Hopf bifurcation. So you have oscillatory behavior. Okay? And so because the equilibrium are all unstable. Okay, so if I put my model into this oscillatory behavior, this is what it looks like. Hey, it's oscillatory. I mean, I know that's straightforward, but one of the interesting things about it is these oscillations all synchronize, and they all synchronize because of the diffusion. Okay, and so the cell goes through these periodic extensions and retractions and the like. Okay, now that's a nice prediction. How do you test it in a real cell? Remember this, if you want to make a real impact in biology, you actually have to then go test it in biology. Okay, otherwise you're just playing at a biologist. So the way to do this is through a very clever technique that was developed by a guy in medical school, which is known as chemically induced dimerization. So the picture is like this. You have these two proteins whose names I forget what they stand for, but it doesn't matter. And they don't come together. But if you add a particular chemical, which rapamycin, you know that, they come together like this. Okay? So you can bring two proteins together at will. And so the idea would be this, something like this. You put one of the proteins, it doesn't matter which one, on, for example, something that you're trying to regulate. You put the enzyme, you attach it to the other protein. Okay? And so when you dimerize them through the addition of rapamycin, you bring the enzyme to its substrate, and then you get a chemical reaction. Okay? And so essentially what you're doing is you are creating these instant mutants. And this is just a picture of something where we've done that, where we have a cell, and what we did is attach this to a protein that's only found in the nucleus. And so what happened is that we actually took an inhibitor. By dimerization, we moved the inhibitor into the nucleus, away from its target protein. So this was actually working backward. And what you see over time is the cell becomes a little bit, this brightness tells you it becomes a little more active. Okay, so it's hard to see in this light. Now I should point out that we did this. The one drawback of this is it's not reversible. Okay, once you've added the chemical, they dimerize and that's it. We now can do this optogenetically. So basically, by shining a laser, that you join them, you turn off the laser, and they come up apart. Okay, so it's really cool. I don't think I have any movies of that. Now, if you're going to do this experimentally, you can't use your Fish-Nagumo toy model. You actually need to know a little bit about biochemistry. This is a simple model of the biochemistry. I won't go through it, but all these numbers basically refer to different loops. And the biochemistry shows that same positive feedback, negative feedback loop that I talked about. Okay. So, okay, just to point out that our models told you that if you knocked it or you got rid of this, it's not a protein, it's a lipid, then you get more activity. Okay. So this is the experiment. That's more interesting to watch movies. So now we just added rapamycin, and if you look at this cell in red, what do you see? That it starts going through these periods of oscillations. Now it doesn't stay, not like my model. It's a little bit more variable, but it still goes through that synchronized periodic extensions and retractions. Here's another movie of a cell doing this. Okay, so if you look at the really bright cell over here, the top right is just shows you that I've actually been able to recruit that protein, and you see that it expands greatly. Okay, and then it will shrink and then go through an expansion again. Okay, and here what I'm plotting is the fluorescence along the perimeter. As time goes on, it gets wider and smaller because the perimeter gets smaller or bigger and smaller. Okay, now you see that this is synchronized with the periods of extension. Okay, and actually this turned out to be a really powerful tool because we could take different proteins and tag them with different. We could tag them. We could tag different fluorescent proteins and actually get a timing diagram. Okay, so in the first one, it's a particular protein called RAS. We measure its activity, and you see that RAS trails by something like 20, 30 degrees the extensions in perimeter. This pH crack is a little more interesting because it lags by 20 or 30 degrees on the way up. It actually leads it on the way down. And this P10 protein, which is one of those back markers that I mentioned, is actually out of phase. Okay, because it's a, what you're seeing is this is a back marker. Okay, so when the cell expands, okay, it all becomes front, and that's why that protein disappears. One other bifurcation is possible. I mentioned about the extension where we could get things really active, and it turns out that this was a little chemically a little bit harder to do, but when you did it, what happened was that the cells actually just expanded greatly, okay? And they basically eventually just tear themselves to pieces, literally, okay? So they become so active that they lose sort of the ability to stay together. And in fact, the collaborators have this as a patent as to how to kill cells. I'm not sure that would is that meaningful. Okay, so we did see when we looked at these cells, we found one interesting phenotype that we had not predicted, which is that cells can actually sometimes activate half their bodies and move really quickly in a very persistent manner. Okay, and in fact, this cell, it's hard to tell, but it's moving about 4 times faster than your average cell. Now, yeah. Yeah, it's a real cell. This is unstimulated. Yep. No problem. So, and this is what the activity looked like. Now, it turns out that to get this, what we called fan-shaped cells, we needed to consider that polarity. Okay. So basically, what happens is that memory system turns on, but only half the cell. And when you include that into our model of a of an oscillatory cell, what happens is, I'll show you again, is half the cell becomes oscillatory and the other half essentially gets turned off. And we were able to get these cells that are fan-shaped, or they look like they're moving sideways. Now, how do we know this theory is right? Well, we found some known mutants are known to move sideways. Okay, and we basically reversed the perturbation. And what we were able to do is actually get them to start looking like normal cells. Okay, so this is these sideways looking cells. Did the perturbation, takes a little bit of time, but after that, they start moving more like amoebas. Okay, okay, so I have 10 minutes. And as I tell my students, the worst thing you can ever do in a talk is go over time. So I'm going to just very briefly tell you sort of things that we've done more recently. So one of the things that we found out is that there isn't one wave or one type of wave. There's actually 2 types of waves, okay? And if you look at these two proteins, okay, so the one's tagged with a red fluorescence, one's tagged with a green fluorescence, they have these cool patterns, kind of like what I showed you before. But if you actually start paying attention to what they look like, what you see is that some proteins have the shape that I've shown you on the right. Okay, so if you plot the fluorescence over space, they have a single peak that's moving. Okay, other proteins have a double peak. Okay, and so if you put them together, basically it looks like that thing's in the middle of this. Okay, now, and the reason for that, and the other thing you can do is, well, sorry, you can, when you actually look at the different patterns, it turns out that they belong to slightly different parts of the regulatory mechanism. So we call them STEN and SEN. STEN stands for Signal Transduction Excitable Network. It's the regulatory proteins. And CEN stands for Cytoskeleton Excitable Network, which is basically the actin polymerization. Okay, and the actin shows this double peak, whereas where STEN shows a single peak. We can also get rid, we know that there exists two excitable networks because we can, through drugs, get rid of one or the other, okay? And we still have the oscillation in there, okay? I'm gonna skip this because, yeah. So this, the other thing we could do is actually do those perturbations that I just told you, okay? Where we would perturb, so in this particular case, what we perturbed was the positive feedback. Okay. made it stronger. Now, our theory says that the velocity should increase, okay? And that, you know, these were simulations showing that the velocity would greatly increase. The experiments showed that it increased, not quite as much as the system, because, you know, there's a lot more going on in the cell than our models. We're not that naive. We could also increase this particular protein. Now, this is interesting because it increases negative feedback to the stent system, but actually increases the positive signal to the send. Okay. And sure enough, what happens in that case, in the simulations, is that the cell becomes hyperactive, but very, very small. So you don't really get much wave. And the reason is because a lot of waves would actually run into each other. And we saw that experimentally as well. Okay. So it looks like more almost oscillatory behavior, but not coordinated. Okay. So those are perturbations we've done. I'll skip those. Now, the other interesting thing is that when we did these perturbations, the cells look different. Okay, sometimes they look fan-shaped, moving to the side. Sometimes they become hyperactive. Sometimes they come like, you know, little spiky things like this. Okay, and we could explain that both in terms of our model about how, you know, basically the size of the wave and also the level of activity, and we could make them be totally dormant as well. Now, all this is done in either dictyostelium and then confirmed in neutrophils. So you might ask, well, how ubiquitous? You know, going back to my title of the slide, how cells move. You know, there are cells that don't move, okay, like your epithelia. Well, it turns out you're right, they don't move, but the same structure exists. And so this is an epithelial cell line, and we tagged the same fluorescent proteins and get, in fact, the same two types of waves: a send wave with a send wave in outer that's enclosing a send wave. And it is actually driving protrusions. Okay, so maybe I'll do this. Okay, you can actually get activity. Okay, so you see those two waves, they ran into each other. So it's an excitable system, the like. That's just showing you that. And you can stimulate it with chemoattractants. So we just added a chemoattractant, and the whole thing, which is in this case is a growth factor, the thing becomes a lot more active. Okay, so the external chemoattractant also changes the threshold of activation. Yeah, so this will be really clear here. Okay, you see that after we added epidermal growth factor, the whole thing becomes super active. So it is here. Okay, you get a dramatic change for that. Okay. What else? So we have the same sort of notion of all or nothing, and in terms of so a small stimulus and a large stimulus give you the same response, whereas a very small stimulus will give you no response. Okay, we get refractory periods like we did in dictyostelium, and we can also do changes using this chemically induced dimerization. And actually, as I was saying yesterday, we can also do this with light now. So here we added a chemical, and the thing became oscillatory, but actually these are much bigger cells. So the oscillation actually takes only about half the cell. Okay. Now, the other thing that is interesting is that we looked at, you know, sometimes your epithelial cells become cancerous, okay, like breast cancer cells. And so we've looked at breast cancer cells and shown that in those particular cells, they are actually showing more wave activity. Okay, and so, yeah, so that's it. So in summary, and I'm not going to let you go just yet. We have all these ways of controlling how the cell behaves, both in terms of activity and in terms of shape, by controlling this threshold of excitable network. Now, what I'm going to finish is sort of what we're doing now. It turns out that if you put, you know, everyone knows that these cells don't live, you know, in a petri dish. They live in this complex three-dimensional matrices and so on. So we've been interested in how cells actually sense this external matrix. And so with some collaborators in University of Maryland, we've been putting cells on these nano ridges. Okay, and it's basically a very small. It's basically a ridged surface. And if you put naive cells there, what you find is that the cells like to move in the direction of the grade there's no gradient in the direction of the range. Okay, so in this particular case, the ridges are vertically, and you see most of those cells are actually moving in that direction. Okay, and there's a sort of pie chart showing the degree of movement. Okay, now that was discovered a number of years ago. We know that mechanics influences the same excitable system. And how do I know this? Well, that experiment that I told you about, where we would give a chemical stimulus, two chemical stimulus some time apart, we do the exact same experiment, but where one stimulus is mechanical, okay, basically by shaking the cell, and the second one is chemical. Okay? And what happens is that we get the same sort of refractory period, that if you stimulate it mechanically, it takes a while before the chemical system can stimulate it. And what that shows you is that these two are going into the same excitable network. Okay, I mean, they're different. I used the letter U to show an input before. So it's two different U's, but they add to the same subsystem. Okay. What that suggested is that we might be able to then control the mechanical behavior of cells, or more accurately, that what the ridges are doing is controlling that input of the mechanical system. So we looked at waves. These are waves of STEN in green and SEN in red in a mechanical ridge. And what you see is that you get waves that are propagating along, longitudinally with the ridges, but you get different behaviors. The red, which is SEN, is primarily on the ridge, whereas the STEN tends to be more inside what I'll call the valleys. Okay. Now, don't get the other thing is they tend to not move over. Okay. Now, don't get a sense that it's because the cell is small and it's in there. The cell is much bigger than the ridges. Okay. And so we have done that. We have been able to show that we can get the same behavior by using the mechanical property or the shape of the ridges to control the threshold of the excitable system. And what we find is that we can make the SEN stay along the ridges, and we can make, sorry, I said it backwards. STEN stays along the ridges. It acts as an inhibitor from these waves moving across the ridge. And so since they can't cross the ridge, what they do is they move in one direction. Okay, so that's work that's in progress. I think that I'm going to finish. So my last slide, except for that, is, one of the things that we're very interested in is this connection between the activity of a wave and shape. And so the idea that I have is, could we use some sort of machine learning algorithms to use wave patterns to predict what the morphology would look like, or vice versa? And if you start thinking in terms of cancer, we've shown that the level of activity is a hallmark of metastatic potential. Okay, and so could you do this? And so I have one student who's been looking at that with the Fitsunagumo, where we know exactly what the waves and the threshold is, and so on. Okay. And so far, it's promising, but a lot of work there. So anyway, I thank you for your time. I hope I have met my goal, which is to make you think that this is kind of cool, even if you might never want to do this. And so thanks for the people in my lab, and primarily my collaborator for over 20 years, Peter Gavriotis, at the School of Medicine, and some of his students and postdocs. So it's exactly 12 and food is out there, but I'm happy to take some questions. Thank you.
I believe that because of the chemicals more like just like trying sitting in front of so I agree with more like and then I was wondering maybe you're right to some computer like that. I think that the cell has to be I'm saying like basically that's making some level of energy of just basically still alive. And just by optimizing it or solving the support that we never actually send that similar. I don't know if the judgment is possible to provide most likely the same or similar.
So the one thing that we did in this, and actually Sanjeev helped a lot when we were thinking of this, is ask the following question. Okay, and it was a information theoretic approach to the gradient interpretation. Okay, you are a cell, your job is to follow this gradient. Okay, so what sort of system would you create in order to follow the gradient? Okay, and it was very interesting because we came up with two solutions. One of them is where there's some expectation, think of that memory. Okay, and what that suggested is that you might want to bias the location, but you have the counteracting, you still want to measure the gradient. And so what that led to were behaviors like that U shape that you saw. On the other hand, we could take a naive cell, which does, no expectation. I'm going to follow the gradient wherever I want. Okay, and that one, if you change the needle, it goes 180-degree turn right away. So, and what we found that in that particular case, which it's a subset of this problem, we got very behavior that looked almost identical to what the cell did. But and so that was interesting. But the key then was to show that in some respects, what we're interested in is how does this actually happen, and show that in fact what the cell was doing was matching what the optimal predicted. Okay, so it's really what in the end I mentioned that what you really want to do is to understand how biology works. Okay, and so you're constrained in that sense. Okay. Thank you.
Question from any of the students. Yes.
So I have two questions. So the first one is what happens when the cell gets does it transform into dust or is it eaten by the rest of the cell?
Okay, so this is interesting because until about a year ago, I thought once the cell got engulfed, that was it. The cell was killed. Turns out that's not true. So this is what topic of host parasites. So some cells can be engulfed, and that cell might still be alive. So that's almost an aside to your question. But what normally would happen is that if you break the structure of the cell, basically the proteins fall out, and so it would be, it's not dust, but it's essentially proteins that are no longer connected or anything like that. So yeah. So I mean that's what neutrophils aren't looking for food, but dictyostelium, it's looking for bacteria for food. That's what it survives.
Yeah, and the second question is, it seems that you can control only one cell, but in real life you have like multiple cells, right? So how do we address this problem that they are like somehow erratic, right? And you have to control each one of them at the siren moment.
I guess we're not really necessarily trying to control if, for example, the optogenetics where we shine a laser, we can actually move individual cells if we want to. But I'm not sure we'd really want to. I mean, a single cell here doesn't do much. So really, what we're trying to control is really as more of a means of testing whether we understand the system.
Pablo might be asking a different question. Mainly, how do cells coordinate? Like I'm sure there are behaviors where many cells will move in one direction, or grow next to each other at the right place.
Yeah. So I mean, normally it's through chemicals. Okay, they all secrete chemicals. So dictyostelium, I didn't get to it, and I don't know if I I have a great movie of it, but I don't know if it's an old one, so I don't know if it runs on my laptop. Let me just see. Yeah, no, it doesn't. So Dictyostome it's a single-celled amoeba. It eats bacteria, but it's very good at eating bacteria. So what happens is that you put in a field of bacteria, and they will chomp and eat everything. So of course, what happens? They're going to starve because they've eaten all the food. So what they do is that they have a mechanism for detecting that they're running out of food, and they start creating a chemical, cyclic MP, which they secrete, and that chemical. And they secrete it periodically. So what happens with that chemical is it creates waves of the external chemoattractant. And they use chemotaxis, the same technique, to actually aggregate. They can then come together, and we're talking 100,000 cells or so, form a multicellular organism that eventually becomes a fruiting, like a stalk and a fruiting body. And if you make it to the tip of that fruiting body, which is roughly 5%, you can lie dormant. And so, that's how they survive. And then you can take one of those, add water and food, and come to life and go off and break them into apart. But the answer, how do they communicate? Through chemicals.
One more question.
Okay, and I think my question is when you to excite the cells faster. You mentioned that after some time the baby gets it doesn't have to do the energy the cell is spending on that excited state or no.
It's not so much the energy. What happens is that this feedback loop, okay, gets reinforced every time you create a protrusion, right? So it's like a stimulus. But on the other hand, there's a continuous, degradation of that signal. Okay, and it's so stochastically, if for some reason you have no more, you go through a period where you don't much stimulate it, the degradation, which is constant, is going to fade away that lowering of the threshold, and so that's why the cell stops moving and then can start taking a new direction. Okay, so there's, all these proteins and all these activities are being constantly degraded.
Last question. Right, then let's thank Professor Iglesias again. Take the lunch, and then we have our weekly progress.